Okay, we're going to start part 3 of movement. And I'm just leaving this slide in here. I've already talked from this, but just to remind you that I'm assuming that you just watched the TED Talk by Ramachandran. So we can, and you have seen a few studies now examining mirror neurons and some really assumptions of potentially what they are doing for us, what their purpose is. And I think I have one more study before I'm going to ask you what you think. So I'm going to describe a study conducted by Beatriz Calvo Moreno, can't really say her name very well, back in 2005, where she had people watch capoeira dancing, which I'm showing down there on the left. I just got these pictures off of the internet, but what I'm trying to show with them is that it has these martial arts aspects to the dancing. And participants also watched ballet, so being shown down on the bottom right. And then the people who were participants were, some were capoeira dancers that didn't dance ballet, some were ballet dancers that didn't dance capoeira, and some were not dancers. Okay, so these are the results from the Calvo Moreno study, where she was examining activity in the premotor cortex, where those mirror neurons were originally discovered. And if you look at the red bars on this figure, you can see that if they were watching ballet, okay, the ballet dancers had a higher percent signal change than the capoeira dancers or the control, significantly higher. If we look at the blue bars, that is the response to watching capoeira dancing. And the capoeira dancers now have the most signal change and significantly more than the ballet dancers or the controls. And my memory is that ballet dancers watching capoeira, capoeira dancers watching ballet, and controls that do not dance, that those are not, none of those are significantly different from each other. So her conclusion was that these mirror neurons are shaped by experience. So I usually have my green question here, and this is, sometimes it generates some discussion, and sometimes I get a little bit of silence, but I do want you to stop and think, and you might want to pause it, although I'm going to leave that silence in there for you to think for at least a moment about what are these mirror neurons responding to. We've looked at several different studies, whether it was looking at intentions or the that kind of action observation system, that idea, watching somebody perform something that I perform, those audio-visual mirror neurons when we're listening to the peanut crack or the piano. So all of that. And then also, not only what are they responding to, since we're seeing them respond to these various scenarios, but what is their purpose for us? And we might say for other animals as well, since these were discovered in monkeys. Okay, so when students are engaged and really interested in this question, they gravitate towards different answers. And a lot of people say social learning, this idea that we can learn vicariously by watching someone else. Some people feel pretty strongly about that idea of their being involved in our sense of empathy and feeling for other people. And other people talk about the importance of figuring out other people's intentions and our kind of response, how we respond. So this really kind of social, being able to handle our social situations. And I think, so I'm kind of hoping that my pauses here are places where you're thinking about this yourself a little bit, and I hope you did pause it, and that you did watch the Ramachandran, because he's clearly feels that this has to do with, the sophisticated mirror neuron system has to do with our sense of empathy, and potentially hinting on that theory of mind. But one of the reasons I pause here is that I think this is one of those areas that show what an open field a lot of this research is. It's very exciting that just like back when Hubel and Wiesel discovered those simple cells and complex cells, people got very excited about what these meant for putting together visual information. As back then we used to talk about these really feature-driven theories because they had discovered these feature detectors. So we're in the middle of that excitement now with mirror neurons. We're not the researchers aren't sure. We don't know exactly what their purpose is, or and we're finding they're responding to various aspects of watching other people perform these activities. So I think it's a kind of fun thing to think about, kind of like the question of the social pain and how much is that overlapping with physical pain, because it is something that they are still trying to figure out. I always think that's really exciting. Okay, so Callett has taken this out of his textbook. It was in a previous edition. And my Sensation and Perception book also had a discussion of this from a previous edition. And I've kind of put them together since both textbooks had different researchers looking at different brain regions, which led to these kind of green questions to think about for you all. So the reason I leave it in here, it's just one slide, but it shows really how a lot of this basic research that we've been talking about can be translated into something very applied, where we are working to help people from what we know about how the brain works. So they are working on creating what are called neural prostheses for these people who are paralyzed, who are really are in the locked-in syndrome. So none of their muscles work except to make eye movements, but the motor activity, the brain activity, is still there. It's just not communicating it's not getting the information to their muscles. And so we're looking for devices that can substitute for muscles in some ways. And they're recording from the brain activity, taking that and getting it into a computer, and then the computer can interpret that activity and act as kind of as muscles, like moving a cursor, doing some specific action, so that they can at least communicate with other people. I have here whether or not, so different researchers have used different regions of the cortex to record from. They have tried the prefrontal cortex, and part of it is thinking about the roles that these regions play when we are deciding a movement. So you might think back to a previous PowerPoint. So some researchers have had tried using the brain activity from different motor areas, and then some have used this parietal reach region, which in your book he talks about it as the posterior parietal cortex. In my other book they call it the parietal reach region, so the PRR. Either way, it's basically that the fact that when we are intending to move, and we're thinking about kind of putting together that information of my body and space, that we are getting information, or we see brain activity in this parietal reach region. And it is something to think about. Where would you try, and what do you think probably works better? My memory is that different researchers have gotten this to work from these different motor areas. So now the question, I think, is probably what's the cleanest, the kind of easiest to use, the most precise, all of those things. Okay, and now I'm going to do talk about the corticospinal tracts, these paths from the cortex to the spinal cord. Your author only goes through 2 of them. I've had other authors go through more, and I feel like it can get you can feel pretty bogged down. And I'm just going to, I feel like I want you to just know the names of them and the basics of what areas of the brain, what areas of the body these corticospinal tracts are innervating or are communicating to. So we have a set of axons coming from the primary motor cortex and surrounding areas, and the red nucleus as well in the brainstem, and they are sending their information directly to these target neurons in the spinal cord. We see this crossover in the medulla and these bulges that look kind of like pyramids. So this lateral corticospinal tract is sometimes called the pyramidal tract. So the pyramids, pyramidal, but it's called pyramidal tract because they look like these pyramids. And that's the kind of crossing over to the other side of the body to communicate to the peripheral areas, to the kind of distal regions. And so I usually, when we're first talking about contralaterality, and I did it this semester as well, when I'm touching something with my right hand, I'm feeling that in my left primary somatosensory cortex. And when I get up and wave my right hand, I'm using my left primary motor cortex. So again, lateral corticospinal tract, remember that's from away from medial towards the sides, and so that makes sense that it's lateral, and I'm using that to control these more distal, my hands and my feet. The only other one discussed in CALT is the medial corticospinal tract, and again, remember medial is now towards the middle or towards the center. Here we have the axons that are coming from many different areas of the cerebral cortex, as well as the midbrain tectum, the reticular formation, and the vestibular nucleus. And now, as it's medial, it is controlling the middle, more of the middle part of my body, so the neck, the shoulders, and the trunk of my body. Axons go to both sides of the spinal cord, and again, I hope that kind of makes sense to you, because when we are moving let's say the trunk and the neck and the shoulders of my body, that's all going to be happening together. So that information wants to be shared, as these are bilateral movements. So usually this is where we break in class, and I give students the 3 of these Pong kind of balls that are these light plastic balls that are really difficult to juggle, just so you know, even for somebody who juggles a tiny bit, where I lose it after a few throws, but I can juggle these kind of bean bags or small stuffed animals for a teeny tiny while. Those little plastic balls are really difficult to juggle, and so we're basically just chasing balls around the classroom for a little while, and I'm going to miss doing that as we are social distancing, because it's funny and it's a break and it gets people up and moving around in the class. But I'm going to suggest you do something at home. If you juggle, you might do that, but I'm going to assume most of you probably don't juggle and don't have something around that is fun or comfortable to juggle with, like something that you know you can throw up in the air and catch and goof around with like that. So I'm going to suggest that you go choose 2 different objects, one lighter and one heavier, and then take each object and just throw it up in the air a little bit and catch it. So I'm doing this with my remote control, just throwing it up in the air and catching it. Okay, but it's good to get, I think, for the discussion, one that's lighter and one that's heavier. And I want you to think about what you've learned so far about movement, because this is what, so now I feel like, other than telling you to perceive the world and how can you apply some of this scientific information to your perception of the world, which we did in a discussion assignment, with movement, you can go move, and you can think about how these different regions of the brain are active, and what kind of corticospinal tract, and so forth. So ask yourself what mechanisms are at work here as you're throwing up one object or the other and catching it. What role are those various motor regions playing in that? And sometimes you might have some questions, but if you do, think, well, I have a target, so I'm assuming it's the premotor cortex is involved, and so like that. This corticospinal tracts and how those are active and what roles they're playing, as well as the proprioceptors and which kind of muscles are being used for this activity. And so I have these green questions. I'm actually going to I think on Monday I can come back to some of my questions so that I can get you all, I hope, a little more engaged in the class, as most of it has been me lecturing, and I do like to have you guys engaged. So I'm going to probably either start with this, or it depends on where I end up today. But we're definitely going to come back to this, and you guys can share some of what you decided to throw up into the air, as well as how you were applying some of what you've learned so far. Okay, I'm going to talk about the basal ganglia a number of times here in trying to explain and expose you to the complications of the basal ganglia. This is a large group of subcortical structures in the forebrain that are involved in movements, as we'll see really the initiation of movements where we are internally guiding these movements. Okay, and people include in the basal ganglia at least the caudate nucleus, the putamen, and the globus pallidus. But usually, oftentimes, they also talk about the substantia nigra, which are these dopamine-producing cells that provide input into these structures. I talk about it as somewhat separated, but some people include it as part of the basal ganglia, as I think of it as the sort of beginning of the chain. And many people also include a subthalamic nucleus. So I have that discussion, or I have the picture there of the thalamus as being part of this. The subthalamic nucleus is the underneath the thalamus kind of nucleus, and then we have the midbrain areas of that is referring to two regions of the substantia nigra. So what I am including here is the direct pathway, and I do this again at the beginning of talking about movement disorders because understanding the basal ganglia is really important for understanding what's happening with these movement disorders. So when I decide to make a movement, my cerebral cortex is active and it is communicating to the striatum. Okay, and the caudate nucleus and the putamen, those two structures, they are often referred to as the striatum. Okay, so that's the striatum. And then the caudate nucleus and putamen, they are sending inhibitory messages to the globus pallidus. And the globus pallidus is sending inhibitory messages to the thalamus and to one of the areas of the substantia nigra. Okay, so that's kind of the beginning of noticing the complication of what I'm doing. Okay, and the thalamus is sending excitatory information to the motor areas of the cerebral cortex and the prefrontal cortex. So what I'm doing when I decide to make a movement and I'm exciting that striatum is I am inhibiting an inhibitor. So normally what we're doing is inhibiting the thalamus from having this activity where the thalamus likes to chat and it likes to talk to the motor cortex. So we would be moving a lot all the time if we didn't have the globus pallidus inhibiting this thalamic kind of communication. Okay, and so when we consciously decide to go make a movement, we are going to inhibit the globus pallidus so that the thalamus can communicate to the motor cortex. Okay, so that's the beginning of the sort of complications of the basal ganglia. I go through that again. That is really talking about the direct pathway, whereas the indirect pathway, there is an indirect pathway as well, but then I feel like I have to explain the two different areas of the substantia nigra, and it gets complicated because the indirect route is really involved in inhibiting the cerebral cortex so that we're not, again, making movements that we don't want to make. And these two routes are working together, again, so that we're not making movements we don't want to make, and when we consciously decide to make a movement, we can make that movement. So I'm saying a lot of that here now with the slide that has words instead of pictures. There's a lot of spontaneous activity in the globus pallidus. I actually didn't say that. So it is inhibiting that subthalamic nucleus, the thalamus. The caudate nucleus and the putamen are basically telling the globus pallidus which movements to stop, inhibiting, its inhibition of. And the basal ganglia, basically we are selecting a movement by ceasing to inhibit it. That's the kind of overall view of what I just explained with the picture. Your author goes through a number of studies that are showing how the basal ganglia, what role they're really playing in this self-initiated kind of movement. And I'm just including two of these. One is the Turner and Anderson in 2005, examining monkeys where, let me see if I get this right, a light came on, and between 1.5 seconds and 3 seconds, the monkey had to move its hand, and it had to get to the spot where the hand needed to move to in order to receive food. The monkey could not start before 1.5 seconds, or he or she did not get the food, and they couldn't finish after the 3 seconds, or he or she wouldn't get the food. So this is deciding on the timing of that hand movement, and that is showing activity in the basal ganglia for that really self-initiated kind of movement. And here are some people whose names I'm not going to say correctly, I'm pretty sure. Wepner and Weiler in 1998. They used PET imaging to look at people's brain activity as they were drawing lines on a screen with a computer mouse. And if the person was drawing a new line on the, or sorry, circle, they were drawing circles, that should say circles, on the screen with a computer mouse. If they started a new circle, we see activity in the basal ganglia. If they went and traced over a circle that was already on the screen, there was not so much activity in the basal ganglia, showing that the basal ganglia are critical for initiating an action, but not when an action is really directly guided by a stimulus that's out in the world. So here's my not so much of a summary slide, but here's a summary slide of where we've been and where we're going, and I hope I'm going to not confuse you with the movement disorders PowerPoint, because I have already saved it as the MP4 file, and I've already created it, and it refers to things that, so I think I tell you to go ahead and watch the two links that are in there of Michael J. Fox and then the man who has Huntington's disease, and I'm going to save those for Monday. So I'm hoping you're watching this, if this semi-summary, that's not really a summary, but so as we're talking about the brain mechanisms of movement, this part of CALIT, we talked about several motor areas of the cortex, of areas of the parietal lobe, an area of the parietal lobe that is also involved in movement. All of that's all those mechanisms that are in the cortex, but also now we've discussed the basal ganglia, and with the movement disorders, I'm going to roll through that again for very briefly, and also give you a link in case you want to hear all that again from somebody who draws really quickly, and it's kind of cool to watch if you're into this kind of thing. And then on Monday, we'll finish up with the brain mechanisms by going over the cerebellum after answering those questions from earlier and watching the two people who have the different motor disorders or movement disorders and talking about them for just a little while. We'll finish up with the cerebellum. And then if we have time, which that I feel like sounds like a lot, although I think the cerebellum, I'm going to keep that shorter than I usually do so that we can move on into wake and sleep cycles.